very sorry to torture you with that history, but I assure you it will be worth it. And I'm going to leave you with a little bit of a warning for my talk, okay? Next generation sequencing should now be, I think, qualified as a gateway drug. Because what you will see is this history here started with me sequencing mouse and human here, and eventually we started doing pufferfish, and pufferfish had some toxins that people in Japan like to get high on, and that rolls into cannabis, and who knows, maybe you'll find yourself strapped to your HiSeq one day with a needle in your arm doing sequencing the poppy plant. But I assure you there's a good medical reason to do this, and so let me just roll you through where we've come from this. So I did start back here at the Whitehead, fortunately, Eric Lander tolerated me for 4 years here, and I don't know why, but we had a lot of fun building the sequencing pipeline, and we've already been through a lot of this history, but we did start working on the cannabis genome back in 2011, and it's still a big mess. We're still trying to fix up this genome. It's 65% AT, it's about 1% polymorphic, it's 800 megabases, it's diploid, and it's in about 60,000 pieces right now. So we're trying to deploy some PacBio work, and we're going to touch on that towards the end to try and clean this up. So there is a good reason to decentralize things in the cannabis field. So decentralization is this new term that's kind of like democratization, except democracy is now frowned upon, I guess. And so people like to actually distribute the power systems, if you will, in the cannabis industry. We are going from a world where there's it's illegal federally, although it's actually legal in Washington, D.C., which is quite ironic, to the state-based system where we have different laws in each state that are a laboratory of democracy testing out whether cannabis is going to work under certain regulatory schemes. It is still impossible, or illegal, I should say, to move cannabis flowers across state lines, okay? So what this means is people end up building labs, brick and mortar in every single state, because we can't have a centralized lab testing model where you ship everything into something like LabCorp and get it tested. You have to build this stuff somewhat redundantly in every single state. So that favors many technologies that are more portable. Instead of having centralized genome centers like we might be used to, we're starting to see platforms like we have outside from Oxford Nanopore that are eventually going to be in a cell phone, and we're starting to see the same thing happen with quantitative PCR. Tools that can now move to USB stick PCR systems are quite valued in these markets because they're much more distributable. So we started building these tools to service this market, which is quite confusing if you look at a map in terms of how things are legal throughout the world. It does require almost a full-time job just to keep track of this. It's always changing. But the better approach is to actually move the testing to the grows as opposed to the grows to a testing lab with such a dynamic. So we started making DNA purification and PCR tools to help look for human pathogens, plant pathogens, and even do some strain fingerprinting for the field. And we now have hundreds of laboratories using this. It's actually growing quite nicely, particularly in California and Canada, where things are becoming more legal. But these are mostly to look at human pathogens that are in the plant. Very few people have died from cannabis overdoses. You may see some news in the literature about tachycardia or cannabinoid hyperemesis syndrome, but you really don't find many toxic events in the cannabinoids themselves. What you do find is aspergillosis in immunocompromised patients. All right, so you tend to see these spores getting into people's lungs if they're immunocompromised. A lot of immunocompromised patients are using cannabinoids. And of course, there's a lot of epileptic children that are on CBD, and you don't want them to get fevers because a lot of seizures are triggered by temperatures, febrile seizures. So you're trying to avoid any type of food poisoning, any 4 Aspergillus species, which are regulated in California now. Staph aureus and Pseudomonas make sense as well. There are a couple of these other tests that are leftovers from the food industry, and they're somewhat antiquated and probably irrelevant and hopefully will go away over time because they're not extraordinarily informative from a clinical standpoint, but they're still being done. So we have quantitative PCR tools that address all these and that can bring the testing local to the state. Unfortunately, the field has taken some bad habits from the food testing industry, and they're still putting things on petri dishes and counting colonies even though they don't know what those colonies are. The vast majority of these colonies turn out to be beneficial, and they just do total counts, total aerobic counts and total yeast and mold counts. This, from all the sequencing we've done and we've published, is complete security theater. It's basically just a ceremonial process that takes a lot of time, and PCR does a far better job of driving more specificity. So there's one paper I'll point you to that went through this. We actually did ITS sequencing on on the microbiome right off of the cannabis flowers, and then also after they went through culturing on various platforms. And what you see when you do this is that there's a complete shift in the microbiome after culturing. And so the act of actually trying to monitor the clinical risk is altering what you're measuring, and therefore is pretty much a Heisenberg uncertainty machine. It really does not give you any sense of the clinical risk, and in fact, it changes it dramatically. Many of the pathogenic microbes, like Aspergillus, do not like to grow in this environment. They prefer to clump. They create all types of spores. Many of these spores are vaporization resistant, or I should say pasteurization resistant through vaporizers that are out there. And what you see is a tremendous shift where Fusarium had almost nothing before and suddenly takes over afterwards. Fusarium is something that has also been a clinical concern, and it's a plant pathogen. So we've come to realize that this plating stuff is 100 years old, it needs to die, and we should replace it with more modern molecular techniques that are going to be a little bit more portable. The second problem we have in the cannabis field is that there is a nomenclature system that's quite hilarious and is unreliable. We have strain names that whenever one gets extraordinarily popular, everyone else in the industry counterfeits those strains to be the Snoop Dogg strain so they can sell it for a higher price. So if you go into a dispensary these days, what you'll find is there's almost no differentiation in price. Very different than a liquor store. You might find $5 bottles of wine, $200 bottles of wine, because people believe a stag's leap is a stag's leap. They do not believe these strain names are real, so no one's willing to pay more than the same net price per gram. This needs to change because there's patients involved. We have patients going state to state trying to find the same strain, and the reason the same strain matters is that these plants can be they're about 1% polymorphic in a diploid genome, which means it's almost the near equivalence of chimps and humans being able to cross and still have productive offspring. So there's a tremendous diversity in the chemotypes of these plants. The terpene profile and the cannabinoid profiles can be extraordinarily different. Therefore, you can't just, if you're doing this for epilepsy, you gotta be very careful. You gotta know which cannabinoids work for you, and you gotta know which ones are in particular strains. What you tend to find are a handful of people trying to call things sativas or indicas, and I always reference them to this paper, which is this is a really good paper on figuring out when people are talking about bullshit. And that is in fact what that nomenclature system is. We've done hundreds of strains of sequencing. We've done PCA analysis on these. There is absolutely zero correlation with indica and sativa genetically. There may have been at one point a long time ago, but they've been so intercrossed now that the nomenclature system does not track with the genetics. So we want to change this because there are in fact patients on the line. And to do this, you do need to deploy some very good sequencing techniques. It's a very polymorphic genome. So we have been deploying a lot of the things that were invented here at the Broad. SureSelect is one of them. We actually like this as a capture system. And it's particularly important in a genome that has a SNP every 100 bases. If you try to take an amplicon-based approach, and there are some folks in the field selling snake oil, but it's snake oil sequencing. They just use 100 base pair amplicons, and there's SNPs all over their primers, and they get allele-specific amplification, and the phylogenetic trees and the certifications they're making are basically worthless. You really need a system that is insensitive to the SNPs that are in there, and the SureSelect platform does a much better job at that. So we've been doing a variety of whole genome sequencing in the field. We've put several dozen of those public. We also used to do a reduced representation shotgun. We've since switched to SureSelect so we could target 30 critical genes in the cannabinoid and terpene pathway, and we're also working on some genotyping tools that are very portable, which we're going to touch on. So this is the current panel that we use. We cover this cannabinoid and terpene synthase pathway with a SureSelect system. It's only 30 genes. This is the type of problem we're dealing with. It's an 800 meg genome. We did go and try and tile 50 variants in all the different publications of different cannabis sequences that are out there. There are maybe 4 or 5 other companies that are now starting to produce data and putting them public. And the challenge is everyone's sequencing their own pet region of the genome, so it's hard to intersect the data unless you intentionally tile their targets, which we have done. And at the end of this, we blockchain integrate this data, and this is becoming important for intellectual property reasons in the field that I'm going to touch on. Now, this is a demonstration of what this database ends up looking like. Is you end up with a large phylogenetic tree that allows breeders to go and find their strain and figure out what is genetically close to it. This is colored in a Roy G. Biv order. So reds are exact clones of one another, orange samples are siblings of another, and it goes, the genetic distance goes in the color of, or in the order of Roy G. Biv. So this helps them find the exact same strain if they need to, which can be important if you're trying to get the same cannabinoid profile. This also helps them find strains that are very, very distant if they need to breed for hybrid vigor. And there's a host of other tools on this website that help them in the breeding process to actually stabilize seeds. Whenever you have a 1% polymorphic genome, when you try to make seeds from that, you get a very diverse set of different phenotypes that come out of that. So what people tend to do is backcross those until the samples become more homozygous, which we display for them up here so they can determine whether or not things are getting more or less homozygous and become more stable. We also mentioned all the different clones or strains that are close in proximity to them, and then provide them some of the hashing of this data into the blockchain. Now, this is a process known as hashing and stashing your data on a blockchain. It has nothing to do with hash in the cannabis field, so people often confuse that when I give presentations. But the reason for this is to create a fingerprint in a database that's globally distributed that can never be deleted. We could house all of this at our own company. We could get acquired, and that data and the business model could change, and that stuff could disappear in 5 or 6 years. Who knows? The Bitcoin blockchain has a good $150 billion reason to exist in 20 years and is more likely to be there than we will. And so we always put a record of this in that database so that people can refer to that from an intellectual property standpoint and as a time-stamping notary. You can't really go to a federal bank and get your cannabis notarized for a patent issuance. People are filing patents, however. Despite the fact that it's federally illegal, they will issue a patent. You just won't be able to prosecute it. So there are very broad patents that are coming out. This is a patent here that describes strains that make CBD and THC, all of them, owned by one individual. All right? Now, this is probably the most medically relevant strain that's being used for a lot of epileptic kids, and it's soon to be owned by a single individual. This is arguably the natural state of the plant. The USPTO just didn't have evidence that this is the Hardy-Weinberg equilibrium of the plant in its natural state. It's only due to prohibition that we bred it to only make THC. Arguably, that it would be a more patentable event because it was man-modified to that point. We did, however, put a type 1 strain, a THC-only producing strain, public in 2011, and that's hopefully a prior art against something like that happening. And we're hoping that people that are contributing to this database, in fact, will prevent more and more of this from occurring. Submarine patents are probably on their way. So back to a nomenclature system that we need to iron out the diversity in these strains. This is probably obvious to everyone here. You basically sort your SNPs for the most impactful variants that are out there, high information content SNPs, run this through a PCA analysis, and you get a list of SNPs that have the most signature for differentiating everything in your database. You turn that into like an ASCII barcode, and this can hopefully be integrated into seed-to-sale tracking systems that people are currently building on blockchains. Okay, so how do we then go about doing this in the field? There we go. Hopefully I didn't skip forward too much. So to make this simple and work inside of a grow, we started working with the New England Biolabs and Icahn Chemical to develop LAMP assays, which are colorimetric PCR assays that just change color in the event that it finds a genetic marker. In this case, we're actually tracking male and female genetics because the actual male plants, no one wants them around. They actually want to get rid of these things as fast as possible because the male plants actually seed up the female plants. And the female plants produce the flowers and produce all the cannabinoids. So it takes about 6 weeks for the plant to express XX, which is about half the growth cycle under lights. There are places that have millions of square feet of lights, and they don't want to waste half of that with growing male plants. So what you find is a lot of cloning going on in the field and occasional breeding to make new lines. The cloning can be a little bit dangerous because you end up with monocultures that are subject to some disease. So we can utilize these tools not only to look for Y chromosomes in the plant, we can utilize them to look for the BT-B allele. We have assays that look for whether the plant's going to make CBD, THC, or cannabigerol, which is a third cannabinoid. This took a lot of PacBio sequencing to sort out the loci and understand which variants are driving this. We have male versus female. These things work in 96-well plates, but they also work in these nice $600 USB stick PCR devices, which are quite portable. So this makes it very easy to get these things in a backpack and out into the field. They only run 8 samples at a time, but you can usually get them down to under $7 a test. It just uses a hole punch, a 4mm hole punch at the plant, and you can get multiple different tests run off of one hole punch, probably 50 different tests if you need to. And you get answers in about 90 minutes, but the most important thing is you don't violate the Controlled Substance Act doing this. A lot of labs are shipping these hole punches or Wattman papers over state lines. We have multiple attorneys review this. You can't do that. The CSA prohibits any plant matter from going across state lines. So this is an impetus to actually move the testing local as opposed to centralizing it. The other issue is privacy. There is the risk that labs who centralize this can be filing IP on the diverse genetics that they find, and if you keep it in-house, you don't need to worry about that. The final thing that we have been developing is tests that pick up all the things that destroy cannabis. Powdery mildew was one of them. We had to sequence this genome because it's novel on the plant. The russet mite genome is another one that we sequenced, and a host of others. So we have assays that now test for all of these contaminants that can wipe out a grow. The grows are, in fact, rarely have crop insurance, and so it's important to screen for these things. And so I'm going to switch gears a little bit here and talk about the blockchain integration that we have. We used to do this on Bitcoin. Bitcoin got very congested and expensive, and we've since migrated over to a faster version of it called Dash that's forked off of Bitcoin back in 2014. All right, this is where we're hashing and stashing all of the fingerprint data so that they have an irrefutable and immutable timestamp for the data. How Dash differs is it only gives 45% of the mining rewards, actually 45% to the miners. The other 45% it gives to another level that's called the master nodes. These are computers that are on the network that have enough compute to actually hold copies of the blockchain, but they also must stake at least 1,000 Dash on the network, and this is about $500,000 now worth of stake that has to be there. That's used to facilitate microsecond transactions that are faster than the block cycle, but it also gives them another 10% that they put toward a treasury, and this treasury can then be voted on by these master nodes what to do with it. And now they are starting to award this to various journalists. They have a free press. They're awarding this to some scientists like ourselves. And they just recently gave us a grant back in December for about $100,000. This was very exciting to do. We submitted this like on December 23rd, and we had cryptocurrency inside of our wallet January 3rd to spend. It was the fastest grant cycle I've ever had. The review process went through in about a week. So it's all online. It's all transparent. You get to see everyone critiquing your work in public. It's a little bit embarrassing sometimes, but I think it's the future. So they're going to sponsor CanMed. They paid for us to migrate our blockchain over to theirs. It's a very similar blockchain as Bitcoin. And they're sponsoring now the scientific conference that we're holding out in UCLA. This is CanMed. It's about 30% physician attended. We give out CME credits to physicians to learn about how to use cannabinoids. It's about 1,500 people. It used to be over at Harvard Med School, but it doesn't fit there anymore, so we have to move it to UCLA. This made us think about what else can we do with this? I think there's enough time to go to the next slide. If you can get cannabis research funded this way, what about publishing, right? We have a publication crisis in the world right now. We have a replication crisis, basically. Half the papers don't reproduce themselves. You have to ask why. What is it about the peer review process that promotes this? The other thing we find is that the type of retraction is often compromised peer review. That seems odd, considering it's all anonymous. And the other thing is the papers that have the highest impact score have the highest retraction rate. All right, so when you look at this from a cryptocurrency perspective, you look at the game theoretics and the economics, and you realize the economics are no longer fit the modern world. The economics were there for a Gutenberg printing press era where it was expensive to print paper. It no longer is expensive to publish. Why do we need journals to do this when it feels like this, right? We effectively give our work to the journal for free. They take the copyright, they take $3 grand, and they charge you $50 to get your own data back. The incentives are completely broken. We have a trust system now that's holding $300 billion worth of cryptocurrency that's cryptographically verified and is all over the world and has never been hacked. We should publish on that. And we should cut these guys out, and we should put out crypto bounties for peer review. That's what we're going to do. And we're going to do this on a cannabis genome, the one that's been patented in particular, so we have a reference that people can find if they're plants that predate 2013. They can sequence those with us. They can match it to that reference. If it matches, they're outside of the patent. So this is the Dash voter network live today voting on this project as to whether they're going to fund it. Now, it looks like they're going to fund it. We're going to put out a crypto bounty probably around October. A crypto bounty is basically we will pay you the cost that it takes to publish to review it. As long as you put your name public and as long as all of your reviews get cryptographically hashed onto the blockchain, you'll get the cash as opposed to the journal. And we're going to experiment with this and see how it goes. We could be wrong, but we think this might improve the accuracy that's going on in peer review. I'd like to thank all these people that are involved and take questions.
Thank you very much. So that's enlightening, right? So any questions for Kevin? There's got to be something going on there. I think you spoke at about 150 miles a minute, so it's hard to keep pace.
Sorry.
It's okay. Any questions for Kevin? How do you guys feel about publications going away? Go ahead.
So I really like the idea of obligations going away, but I think it still doesn't solve a problem, which is like just there's so much out there and things are repeated and you don't know how many times the same thing has been replicated. So that problem, whether it's paid or not, is not going to go.
So I like that point. I think the point is about replication. So one thing that's not going on in peer review today is it's somewhat of a one-size-fits-all review process. If you put a pricing signal on review, you could have premium review and you can have bronze review. You might actually fork over 20 grand to improve, to give to someone to actually reproduce it so that your paper gets stamped with a reproduction credit of some sort. That doesn't happen right now. Papers get out there, you download them, you try them, they don't work, you get frustrated, you contact the author, and you can't resolve this. So I'm hoping that when there's a pricing signal on review, we'll see it speciate a little bit. There'll be quick reviews, there might be bioinformatic reviews that can do in silico reproduction, and there might even be wet lab reproduction on review, and those will get tiered and probably priced accordingly. That's my hope.
Anybody else for Kevin?
So this is a pretty naive question. I don't know too much about blockchain, but who is actually voting to give you the grant money?
So that's the masternode network. There's about 4,700 of them in Dash, and that was that map that I showed of all those Ds. That's where all the masternodes actually exist. We don't actually know who they are. They're just people who have at least about $500,000 worth of this cryptocurrency, and as a result, they have a motivation to fund things that will improve the network and grow the currency usage. So they like this concept because they see there's a banking problem in the cannabis field that they can solve, and so they want more exposure in the cannabis market.
All right. Thanks very much,
All right.
Kevin.
Thank you.